Pedro, okay? So, Dr. Tokuyama is an assistant professor at the University of British Columbia in the Department of Microbiology and Immunology, where she's just opening her lab starting early this year. She's interested in the interaction between viruses and the immune system, and she started looking into this kind of into this field as an undergrad, where she started working with HIV research, and later she did her PhD in molecular and cellular biology at the University of California in Berkeley, where she studied how immune cells kill cells infected with herpesviruses. After that, for her postdoc, she worked at Yale University in the Department of Immunobiology, where she became fascinated by endogenous retroviruses. Dr. Tokuyama's current research focuses on understanding how these endogenous retroviruses affect the immune system of their hosts during health and disease. The past year, she has also been helping, and she has also taken part in the Yale Impact team to develop a rapid saliva-based qPCR assay for SARS-CoV-2, and she was also doing research on the immunological impacts of COVID-19. So, Dr. Tokuyama, thank you very much for taking the time to be here with us. I apologize for having to make you available so soon for you, so early for you, sorry, and it is a great pleasure, and I wish you an amazing talk.
Well, thank you so much, Fabio. Thank you so much, committee. And thank you all for inviting me and giving me this opportunity. This is exciting. I've never been introduced in Portuguese before, so there's a first for everything. So I'm going to go ahead and share my screen, and please let me know if there are any issues. Okay.
It's rolling. Just 1 second. Yeah, we can see your screen. We are now on PowerPoint. Perfect.
Great.
Yes.
So again, thank you all for this invitation. You know, in this time of COVID and challenges and difficulties associated with the pandemic, sort of despite that, the silver lining is that we can connect with each other over Zoom and video in this manner. So this is really fantastic, and I'm really excited to share my research and my work with you. As Fabio mentioned, I am currently an assistant professor at University of British Columbia, and I just wanted to give a quick land acknowledgement. As scientists, you know, working in the University of British Columbia, we live and learn on the traditional and ancestral and unceded lands of the Musqueam Nation of the Coast Salish peoples. And so I just wanted to give that acknowledgement up ahead and share with you our research. Okay, so we all know, and you are all very familiar with the fact that our bodies are actually outnumbered by microbes. So collectively, we have a collection of bacteria called the microbiome, and this consists of trillion cells and roughly 1,000 species. And I think we can now say, you know, very confidently that the microbiome actually affects everything in our physiology and in health. And really, you know, the last couple of decades have really illuminated that the microbiome is really important for everything, including pathogen clearance, inflammation, development of autoimmunity, cancer, metabolism, aging, and beyond. And so, you know, this is very well established, and many investigators are you know, doing really great research in this field. What is sort of the less well-known cousin of the microbiome is the virome. So the virome is, you know, in parallel to the microbiome, except these are viral components. And the virome consists of viruses that many of us actually carry and harbor currently as you listen to this talk. So these are viruses that, you know, you have been exposed to at some point in your life, during childhood, during adult life. And it, you know, a well-known example is a herpes virus or herpes simplex virus, like the cold sore virus, right? You've been exposed to these viruses throughout life. and they actually remain latent in all of our bodies. So they do not cause overt disease on a day-to-day basis, but we, carry these viruses with us for life, and this consistent, persistent interaction between these viruses and the immune system can potentially impact our health in much the similar in a similar manner as the microbiome. And so the human virome is composed of again a number of viruses, and what we're really interested in understanding is really how these viruses and the viral components interact with immune cells, and how that in turn affects antiviral, antimicrobial responses, or how we respond to chronic inflammation. And so this is this idea really comes from Dr. Herbert Version at Washington University in St. Louis. Their lab has been working on this for a very long time, and this, our research really has been driven by the excitement of his research. So just to give you an example, one of the work that has come out of Skip Version's lab modeled this interaction between gamma HV68, the immune cells, and then how you respond to Listeria monocytogenes. So this was all done in vivo in mice, and what they were asking is, does prior infection by gamma HV68 affect the outcome to Listeria monocytogenes infection? And so here what you're looking at is mice infected with gamma HV68 one day prior to Listeria monocytogenes infection. And you see that looking at the survival curve, there's no difference between mock infected or gamma HV68 infection in response to Listeria here. But what you can see here, and what's really amazing, is that if you infect mice 4 days before or 12 weeks before with gamma HV68, and then come in with Listeria monocytogenes, then this persistent interaction that the immune system, the persistent interaction between gamma HV68 and the immune system really gave these mice the survival benefit when they were infected with Listeria monocytogenes. And, this paper revealed that this was dependent on interferon gamma, and there were other mechanisms that this paper revealed. And I think that this is a really great evidence that a prior interaction between sort of the viral components, persistent viral infection, way past its clearance time, and the immune system can really impact how the organism responds to secondary infection or secondary stimuli. And so we're really interested in this idea and focusing specifically on how endogenous retroviruses, or ERVs, which are at the top of this list in sort of the contribution to the human viral metagenome, and how these ERVs interact with our immune system, and how that impacts our health and disease outcomes. And so just to give you a couple more examples, this is not, there are many examples that have been shown in the past where herpes viruses or prior virome component viral infections can lead to differential outcomes, or even cause some of these diseases. And so, for example, cytomegalovirus has been shown to affect outcomes to influenza. There's been a long-standing study looking at interaction between prior herpes virus infection and seropositivity to HSV, to HIV, so on and so forth. And so this idea of a viral origin of disease outcomes is not something new, but we're really trying to understand the mechanism of this of the viral origin of these diseases. And so so we focus on endogenous retroviruses, and so what are endogenous retroviruses? Well, our human, all of our human genomes actually consist of 8% of our genome consists of endogenous retroviruses, and that's present in all of the human genomes. And so unlike other viruses in the virome that you have to be exogenously exposed to, endogenous retroviral sequences are actually already present in all of our human genomes, and they belong to a larger class of sequences called the transposable elements. And this collectively with ERVs, DNA transposons, lines, and signs, they actually make up 40% of the genome. And this is really in stark contrast to 2% of the genome that encode for protein, which, you know, we have based all of our understanding of our biology and health and disease on to this date. And I think what we're interested in is really trying to reveal more about the functionality of these endogenous retroviruses and transposable elements in health and disease. And so when the human genome was sequenced back in 2001, Eric Lander and collectively Eric Lander and many institutions and investigators published this report revealing the composition of the human genome. And one of the things that in this paper, in this 2001 Nature paper that they published, they mentioned, and I kind of wanted to bring this up here because I think it's you know, really revealing of how little we know about these sequences. So repeats are often described as junk and dismissed as uninteresting. However, they actually represent an extra matter, or 98% of the genome that doesn't code for anything. I think we're really starting to appreciate and starting to slowly beginning to understand how these elements are actually relevant in disease, and not just as fossil records, but I think important for human genome sequencing. And I think we're now at a stage where we can continue and expand upon such work. And so, as I mentioned before, 8% of the genome encode LTR, or endogenous retroviral elements, and that's 4 times more than the genome, the protein coding sequences. And so where do these come from, and what do they do? I wanted to give you a quick background on and what we think, where we think they came from, and you know, some known, some research on what we know about these sequences. And so we think that these retroviral sequences arose in the human genome initially through exogenous infection by retroviruses, very much in a similar manner as HIV infection, except this happened, you know, in our ancestral genomes millions and millions of years ago. And so, as you know, retroviral replication cycle includes this step where the viral RNA is reverse transcribed into viral DNA, and this double-stranded DNA is integrated into the host genome. And once it's integrated into the host genome, it can actually be transcribed similar to any other cellular genes and generate protein. And then this leads to propagation of retroviruses. And so, you know, this integration step is key to the retroviral genome, and it's very unique, unique to retroviruses. And so there's studies, and based on, you know, many investigators' research, we know that this integration event must have happened millions of years ago in discrete times in our ancestral genomes. And so different families of ERVs that we now have, ERV sequences that we have in our genome have been, we think, originated from different points through evolution. So, for example, the peak invasion events we think happened 30 to 40 million years ago. However, there remains some endogenous retroviruses that we think integrated specifically into the Homo sapien genome. And so these are the ERV-K family of ERV sequences, of retroviral sequences that are purely Homo sapien specific. And I'll kind of come back to this later. But we can see that, integration events happen through evolution, and this is what we've carried. And so carried through evolution. And so just sort of an idea of how this could have happened and would have happened is various integration events occurred in the genome of our ancestral, our primate ancestors. And in some of these, obviously, integration events, did not survive through evolution, right? And did not get fixed into the genome. However, there are lines of retroviral sequences that have that became fixed and then eventually got inherited through generations, and that's what we see sort of today. And so these sequences that we see today are those that have survived sort of through evolution and have become fixed into the genome. Okay, so when this initial integration event occurred, these, similar to other retroviral sequences, they integrated into our genome as proviral sequences or proviral ERVs. And again, similar to other retroviruses, these are the structure of these proviral ERVs is as such here. So there are two flanking LT sequences on both the 5' and 3', and that is flanking internal sequences that actually encode for viral proteins like GAG, POL, and envelope. However, through many generations in evolution, through many generations, these proviral ERVs have actually acquired large deletions, and also many of them have undergone homologous recombination via the LTR, the repeats, the long terminal repeats. And so most of actually the sequences, ERV sequences that are remaining in the human genome take the form of solo LTRs, so just a single LTR that arose from homologous recombination between 5' and 3' LTR, or internal sequences that have been that has resulted resulted from large deletions. And so, however, there are still remains proviral sequences in the genome that do still maintain this proviral structure with LTR, and viral proteins in between. So just to give you an example of what these LTR sequences do functionally, so what we know is that these ERV LTRs, or at least these solo LTRs, are really efficient regulators, cis-regulators of gene expression. So as a long terminal repeat promoter, if they lie upstream of a known protein coding gene, a cellular gene, it can actually serve as alternative promoters and enhancers. And this has been shown for many genes now. And these are just examples of genes that I'm just highlighting here. So what does this actually look like experimentally? Just to give you an example, so this was a beautiful work done by Cedric Fischelt's lab back in 2016, where they actually looked at this miR41 LTR element that's upstream of this gene AIM2. And they found that this miR41 LTR sequence encoded a STAT binding site that is responsive to interferon gamma. And so when you treat cells with interferon gamma, you see that AIM2 expression goes up. And what was really exciting was that when they deleted the miR41 LTR element that's upstream of AIM2, AIM2 was no longer responsive to interferon gamma. So really suggesting that these LTR elements can serve as an efficient promoter for genes. And on the flip side, sort of proviral ERVs have also been shown to have various functions, important functions in biology. And here are some examples. So proviral ERVs that are transcribed as similar to cellular genes as polyadenylated transcripts. They can actually form long non-coding RNAs to regulate gene expression. And this has been shown in the case for embryonic stem cell development. We also know that these polyadenylated transcripts proviral derived from proviral herbs can generate viral proteins, including GAG, pol, and envelope. And so, and just to illustrate this point, so these, virally encoded proteins are, by nature, they're supposed to encode for example, envelope is supposed to be to form the envelope of virions and viruses. But in this case, these proviral sequences are actually integrated into the genome. So instead of forming virions, they can also further be expressed on cells that can transcribe this region, encoding the envelope. And so not only are they viral envelopes, but now you can have these viral proteins expressed on cells and can have functional consequences there. And so one really well-established example of this is syncytin 1, which is actually derived from an ERV-W envelope protein. And so syncytin 1, you can see here is highly expressed in the human placental tissue. This is negative control, and this is stain for ERV-W envelope. And what we know from various studies, including those listed here, is that syncytin protein is really important, and they're really important for this interaction between the maternal and fetal blood. And so they're expressed by syncytin trophoblast, and it really provides this nice structure, this ST1 and ST2 structure that forms a really nice epithelial barrier between the maternal blood and fetal blood to have really nice exchange between the fetus and maternal blood. And so this is one really good example of a functional envelope protein derived from endogenous retroviruses. And ERV-W has further been shown to, independently of this phenotype, have been shown to modulate the immune system. They've been shown to activate monocytes through TLR4 engagement. Some ERV envelope proteins encode an immunosuppressive domain in the transmembrane region, and that can actually have impact on T cell and B cell function. And furthermore, although more rare, there is evidence that proviral ERVs can lead to production of viral-like particles. ERV proteins can be present on exosomes, and they've also been shown to have antiviral function as sort of truncated envelope proteins. And so, although our understanding of ERVs and biology is still at its infancy, there's really growing evidence suggesting that these are clearly not junk. They're clearly not just sort of genomic fossils, and there's more sort of beyond what we've originally thought of as important function of ERVs, and I think there is more function associated with these ERVs that's important for biology. And so, that's sort of in the absence of disease, and we actually now have a lot of evidence suggesting that ERVs are highly dysregulated in various disease settings. So neurodegenerative diseases like MS, ALS, schizophrenia, bipolar disorder, in autoimmune diseases like MS, SLE, type 1 diabetes, and RA. And we also know that in various cancer settings, ERVs are also dysregulated, and they are being sought after as sort of new tumor-associated antigens that can be targeted. And so I think there's growing evidence that there's dysregulation of ERVs in disease, and I think our understanding, what we're still trying to understand, is the mechanism of how these dysregulated ERVs contribute to disease pathogenesis, and what their roles are sort of in immunity. In part one mechanism by which these ERVs are dysregulated, we think, is through disruption of epigenetic silencing of ERVs. So again, there's a really great body of evidence and research showing that endogenous retroviruses can be silenced epigenetically by these sequence-specific Crab zinc finger proteins. And Crab zinc finger proteins actually bind to its target in a sequence-specific manner, recruit histone methyltransferases and DNMTs to actually then add silencing histone marks and DNA methylation marks. And so, and this is one major form by which endogenous retrovirus are repressed. And so in cases where there are ERV expression, the likely scenario that's occurring is that there's reduced epigenetic silencing that is then resulting in expression of these endogenous retroviruses transcripts. Okay, so my lab and my research thus far in my lab is really interested in understanding 4503 locus endogenous retroviruses. You can see that the H3K4 trimethylation nicely sort of mirrors this, the RNA sequencing expression data as well. And so that suggests that, again, there is epigenetic control of these endogenous retroviruses, and what we see at the transcript level is reflective of the epigenetic marks that are present at these loci. And this is one example of such phenotype. So we can further use this pipeline to look at disease tissues. So, for example, in this paper, we looked at breast cancer tissue samples from TCGA, and we observed a number of sort of differentially expressed endogenous retroviruses, each dot representing an endogenous retroviral loci. So these are locus-specific expression profile. And what we can do now, because we have locus-specific analysis, and we can compare it to cellular gene expression, per individual, we can see correlation between expression of ERVs and expression of any immune signature. In this case, we looked at cytotoxic T cell signatures, and we can see that there are a number of endogenous retroviruses that correlate highly with, per individual, correlate with cytotoxic T cell signatures, which may suggest some, biologically, this association between sort of ERV expression and T cell signatures or cytotoxic T cell signatures. And so we think that this is a very powerful tool. In the past, there was only 8 ERVs that were identified to correlate with perforin and granzyme in a similar type of analysis, but with a different pipeline. And we've been able to sort of expand this to larger numbers. And so I think we can really start to investigate what these ERVs are really doing. And so so that was that was sort of the summary of the development of ERVmap, and we've now taken this ERVmap tool to look more in detail how these endogenous retroviruses might be playing a role in disease, and the disease that we are focusing on is systemic lupus erythematosus, or SLE, and we chose this disease initially because there had been implications of ERVs in this disease in the past. However, there had been no mechanistic understanding of how these ERVs are potentially contributing to disease. And so we really wanted to use our ERVmap tool to see if we can expand our understanding of ERVs in SLE disease. And so SLE is a systemic autoimmune disease, and this affects roughly 1.5 million Americans with lupus and over 5 million people worldwide. Despite the sort of health burden that we observe with lupus, there's actually only one FDA-approved biologic, and this was approved in 2011, and this is called belimumab. It's an anti-BAFF monoclonal antibody. And one of the biggest challenge with SLE is that it is systemic and very heterogeneous in clinical manifestation. So we know that there are contributing factors that contribute to disease development, including sort of the genetic components, the sex of the individual. So this is a very female dominant disease. It's a 9-to-1 female to male ratio. There are, we know that Asians, East Asian individuals who are Asians or East Asian African Americans actually have more severe disease than Caucasians. Hormones, environment, and sort of age all sort of contribute to the pathogenesis of this disease. And this leads to a really wide range of clinical manifestation that's systemic, right? So you have disease and inflammation that you can see in the skin. This is a very, butterfly rash is a very well-known clinical manifestation of initial clinical manifestation of lupus. But this disease, this autoimmune disease affects all organs. And, you know, when it's not treated and when you can't sort of tamper the inflammatory response, you end up with kidney failure, and one can succumb to death. And so this is a major health issue. And the challenge clinically is that because this disease is so heterogeneous in between and so different between individuals, that one drug can't affect and treat all of these symptoms. And so we really need a better way to segregate these different symptoms, different disease manifestations, and really understand the underlying component of these clinical manifestations, and that is going to help develop better therapeutics, personalized, that's closer to personalized medicine that can actually lead to differential treatments of disease. And so we wanted to apply our ERV map to really investigate whether endogenous retroviruses in our hands and through our analysis is differentially expressed in SLE, and potentially that can have a that can contribute to inflammation in SLE. So we again started with RNA sequencing data. This is data that we obtained through our Yale cohort SLE patients, and we saw with our cohort that endogenous retroviruses are elevated, significantly elevated in SLE compared to healthy controls. I apologize for the colors. So the green is supposed to be SLE and the orange is supposed to be healthy here. We've further confirmed this in a larger cohort of SLE patients, and this expressed ERV loci in SLE patients and in both cohorts. And so the rest of the talk will be on this cohort here. And this is sort of expanding beyond previous investigations that have shown, you know, 2 real main ERV loci implicated in SLE, and now we have sort of over 100 ERV loci to work with. So I think this is a great advancement in our understanding. So then we took these ERV expression data and we correlated with some clinical correlates of disease. So for example, here we're looking at autoantibody levels, autoimmune diseases with patients who have autoantibodies, and specifically in lupus patients, they will have autoantibodies against nucleic acids. So anti-nuclear antibodies, anti-double-stranded DNA antibodies, or anti-RMP. These are all very classic, well-established markers of autoantibody development in lupus. And we see a positive correlation between in the ERVMap database. Roughly 87 are ERV-K family members. And as I mentioned earlier, ERV-K is a Homo sapiens-specific endogenous retroviruses. And within the ERV-K family, there are 12 that encode an envelope protein. And so we focused on these 12 envelope coding ERV-K family members, and specifically found that K102, K110, K106, and K115 are significantly elevated in SLE patients at the transcriptome level compared to healthy controls. Furthermore, specifically focusing on these ERV-Ks, we found that sort of consistent with SLE disease phenotype, that ERV-K102, 110, 115, and 106 are elevated in females. And this seems to be a female, but not in males. And this seems to be a female dominant signature, again, consistent with what you see in SLE. And then we also observed that in particular, K102, 110, 115, and 106 are highly elevated in patients, in SLE patients who also have a high interferon-stimulated metric score. So these patients have high interferon signature scores, and it seems to correlate with the ERV expression here as well. We confirmed that the ERV-K102 that we see highly elevated in RNA sequencing is it is K102 sort of by traditional Sanger sequencing. So we designed a pan-ERV-K primer based on a previous study. Sequences was that SLE patients do indeed have antibodies against ERV-K102 that's represented by both IgG1 and IgG2. And this is compared to sort of canonical, well-established self-antigens like Rho and C1q, or a sort of vaccine antigens that we've all seen, which are primarily IgG1 dominant signature, whereas these anti-ERV antibodies that we see in SLE patients were both IgG1 and IgG2. So further characterizing what these anti-ERV-K102 IgGs may potentially be doing in this disease, we found that they don't necessarily correlate with disease signature or the disease severity. So SLE-DAI is a clinical score for SLE disease severity, and there wasn't a significant correlation between the amount of IgG and SLE-DAI. This IgG, anti-ERV-K102 IgG, actually does seem pretty stable over the course of 6 to 12 months. So there's no really big fluctuation in the level of IgG, within the short time frame that we looked at. However, we did observe that the level of IgG per individual, the level of anti-ERV-K102 IgG per individual did directly correlate with ISG expression or interferon-stimulated gene expression, suggesting that, overall, although it doesn't correlate with disease severity, there is a correlation between the amount of anti-ERV-K102 IgG present in SLE patients and the interferon-stimulated gene expression that we see heightened in SLE patients, as I mentioned earlier. Furthermore, what we found was that when we generated immune complexes with these ERV-K102 antigens to see whether immune complex mediated function can be discerned. So here what we're doing is conjugating a recombinant K102 antigen that's the SLE plasma, feeding them, in this case, looking at neutrophil function. What we found was that these immune complexes that are generated with SLE plasma, so SLE IgG, both plasma and IgG purified, these immune complexes are actually more readily phagocytosed by neutrophils when they are generated with SLE plasma compared to healthy controls. So again, this suggests that there's something functionally distinct about dysfunctionally distinct. The antibodies against ERV-K102 is functionally distinct, despite the fact that it doesn't overall correlate with the disease severity, right? So there's something functionally distinct about these antibodies. So we pursued this question a little bit further to ask how this could be. And what we found was that this enhanced neutrophil phagocytosis score, or antibody-dependent neutrophil phagocytosis that we see, it does correlate with the amount of IgG2 specifically. So as I mentioned earlier, Fc gamma receptor. And so when we take these ERV-K102 immune complexes and determine, how well they bind to various Fc gamma receptors, we found that Fc gamma receptor 3b is actually
it could be an interesting question.
Yeah, no, I think that's a fantastic question. Thank you so much for your curiosity and your question. I really appreciate it. I love this engagement. And I completely agree with you that there might be other sort of serum components, including complement, that could be affecting this effect. I think, although we haven't done an exhaustive investigation on this, we did look at purified IgG. So we purified IgG out of plasma using sort of IgG purification methods, which would sort of eliminate any of these plasma or serum component, protein components. So we still see an enhancement in neutrophil phagocytosis. So although it doesn't I don't think we've eliminated the possibility that other factors are contributing to the overall effect of neutrophil phagocytosis, we have seen that IgG, purified IgG alone is sufficient to induce this neutrophil phagocytosis. But I think you have a really great point, in physiological settings, obviously we're not gonna have purified IgG, right? And so in that scenario, I think it is really something that we can look at. We've actually talked to our collaborators initially about looking at characterizing these anti-ERV-K102 IgGs a little bit better. And one thing that we can actually do is look for complement deposition on these IgGs specifically. And so that is something that we could, definitely do and can contribute to the overall phenotype. I agree with you.
On the structural basis, you don't know exactly what are recommending in this complex, right? Okay.
I don't know. I don't have a structural information on that. Yeah.
Thank you. I'm sure that my colleagues have a lot of questions. Thank you so much.
Okay. Yeah. Thank you.
Thank you, Julio. I'll ask everyone who would like to pose a direct question to Dr. Maria to raise your hand through the application, because my connection is also not very good, and sometimes opening the camera is not enough because I can't see all the open cameras. So we have several messages thanking you and saying that it was a wonderful talk.
Thank you.
And I will call for Ulysses, who's also would like to make a direct question to you.
Yeah.
And thank you again, Ulysses, for making the bridge such that we had Dr. Maria here to give us a talk.
Maria, congratulations. It was a wonderful talk, very informative.
Thank you.
And I think that people are thinking about the models and the role of herbs in several situations.
Mm-hmm.
So I was thinking about the model of lupus that you present and the expression of the RFK.
Mm-hmm.
And was thinking about the situation. You think that a chronic inflammation, like in lupus, would lead to epigenetic change?
Mm-hmm.
So making the ERV transcriptional curve,
right?
Or could it be some event?
Mm-hmm.
Event, then you have this ERV transcription,
Mm-hmm.
and then a sustained inflammatory response,
Right.
mediated by innate response and so on. So what do you think?
Thank you, Ulysses. This is again, thank you for this opportunity. I really appreciate it. And I really enjoyed connecting with you over Zoom this past 6 months. And this is a great question. I think it's still, I think one evidence I have is that when I treat human PBMCs with interferon itself, just to ask the question of whether the inflammatory trigger can induce ERV expression. That alone doesn't seem to be sufficient in our hands to derive ERV-K102 or these ERV-K induction in healthy PBMCs, right? And so although, I think it's hard to do the experiment of sort of then, what would happen if you also, in addition to this, have already an epigenetic dysregulation, and then on top of that come in with sort of an inflammatory stimuli, then would just see an induction of ERVs, because you might need to sort of prepare these sites to be open first before the inflammatory stimuli comes, right? And we can't really mirror that in healthy PBMC. So it's possible that the experiment can be optimized a little bit more. But that with data would suggest that sort of inflammatory cues alone is not sufficient. I think there is a combination of these, both a sort of an epigenetic component and inflammatory component that could be involved in the induction of herbs. Yeah.
Okay, so if I may make another question?
Yeah,
sure.
Okay, so if I'm not wrong, the ERV-K expression is sustained for a long time. I think that was for months. So it seems that there is no negative regulatory loop in the epigenetic control of expression. So what do you think about what's happening in this scenario?
Right. So the data for the longitudinal data that I showed you was actually looking at the antibody response. So those were looking at the antibodies against ERV-K102, and the antibody titers don't seem to right? And I haven't looked to see whether there's sustained ERV-K expression at the transcriptome level. That's actually a really great point to kind of look at whether there is an on and off switch. Although, you know, if it is sort of kind of following up with your former idea of whether inflammatory stimuli can sort of then drive expression, there could be multiple factors that are contributing to this expression, and if sort of inflammatory milieu is in part contributing to the expression of ERV-K, which I don't have a definitive answer for that yet in SLE, but if it is, then you can see that you would imagine that sort of there wouldn't, you know, unless there is you know, drug treatment or anti-inflammatory treatments that are being externally made, you might see a sustained expression over time. Yeah.
Okay. Thank you very much, Maria.
Thank you, Ulysses. We have a question from Fabio, who's not being able to open his mic and because we couldn't understand anything, so he wrote it down in the comments here. So he said, I was fascinated by the issue raised concerning the evolution of these sequences in the human genome. I wonder how long they have been there or accumulating.
Yeah, so ERV-K102, or in ERV-K family specifically, integrated into the Homo sapien genome. Some of these are integrated into the Homo sapien genome less than 1 million years ago. K102 in particular is very specific to Homo sapiens. So you actually won't find this in any of our primate ancestors. And so, but, you know, however, there are plenty of sequences in endogenous retroviral, or LTR sequences in our genome that is highly conserved through evolution. And so, you know, for example, insistential 1 is conserved, is highly conserved, and other LTR elements are as well. So it really kind of depends on which ERV we're talking about and which locus we're talking about. But for K102 specifically, this is a human-specific ERV.
Okay, do you happen to find any natural variants or I guess subspecies that don't have them? And do you know if they do exist, if they have evolutionary advantages or disadvantages?
Yeah, so that's a really great question. I don't have an answer, a direct answer for this. So we know that, for example, some of the ERV-Ks, like K115 and K113, there's still an insertional polymorphism between the human population. And so there are different positions even in the Homo sapien lineage, where K113 and K115 seem to be polymorphic in terms of the position that you find in the human genome across individuals. And so, and that's also evidence that this is a recent integration and not a very ancient integration event, but these happened sort of within the Homo sapien lineage, right? And beyond that, we don't know too much about sort of natural variation of these ERVs. I think these studies are being done by evolutionary biologists in this field who are really interested in answering these types of questions, but we don't have a lot of answers yet. Yeah.
Okay, and I'm not completely sure if you mentioned it in your talk because we're dealing with the internet, but do you know if they play any role in transplant rejection or anything related to that?
Yeah, I'm not aware of any studies that have looked at ERVs and transplant rejection, except for in pigs. So pigs are actually, pig endogenous retroviruses, or PERVs, have been studied in sort of CRISPR, and CRISPR method has been applied to sort of delete endogenous retroviruses in pigs to improve sort of transplant efficiency. That is not known for humans, but there is evidence in other organisms that may affect transplantation as well. So.
Okay, thank you.
Yeah.
Do we have any further questions? As I said, we used to have a key that would be open the video and we call for the person, but since the internet has been oscillated, we have to wait for the little hand to raise in case somebody wants to talk. But I think that we don't, and so I believe that I will thank you very, very much, Dr. Maria, for your time and for the discussion that we have had here. I'm sorry for all the internet distress. I'm sure we usually have more participation from the YouTube people, but I guess because of the breaks, they will be waiting for us to post your talk again.
I'm happy to take additional questions from others by email. If anybody wants to email me, I'm happy to share my, you guys have my email address, so feel free to share my contact information. If anybody else wants to have further discussions about this, I'm happy to have additional conversations and discussions beyond this talk. So,
sure, thank you very, very much. And thank you all for enduring with us the problems with the internet. And I'm sure the data helped a lot because your presentation was really amazing. Thank you.
Thank you so much, everyone. Thanks so much for coming. I appreciate it.
Thank you.
Thank you, Maria.
I'm sorry for all the trouble.
No, that's okay. Don't apologize. It's okay. Thank you so much. I appreciate the opportunity. I appreciate it. Hope to see you guys at some point.
Right.
Hopefully we'll be able to invite you again once we are all vaccinated and we are being able to travel and move around again.
Yeah, that'll be nice.
Thank you very much. And thank you, everyone. Obrigada a todos. E nos vemos aqui novamente quarta-feira que vem.
Thanks.
Thank you.